So if you have three targets, if you can produce 3 different guide RNA, then your job is done basically. Now see, this Cas9 enzyme and sgRNA, they are in stoichiometric balance, 1:1. One sgRNA will be loaded by one Cas9 molecule will be loaded by one sgRNA molecule, right? But if you can produce a pool of sgRNA of different types, each can individually be loaded in the same Cas9 enzyme, because this Cas9 is same and the scaffold is also same. Okay, only the difference is 20 nucleotide sequence, guide RNA sequence. So if you can generate a pool of this different guide RNA containing sgRNA molecules, so it can be loaded differently in the different Cas9 molecules and it can target types. Each can individually be loaded in the same Cas9. Okay, so this is it. Okay, now this seems simple, but question is how to do it. How to produce multiple guide RNA simultaneously inside the cell. You have to do it inside the cell. You cannot put it from outside, right? So what are the strategies? So this is the first strategy. Here comes the first strategy. Okay, so instead of this editor plasmid, where it contains only one single guide RNA driven by a single this your RNA Pol3 promoter, what you can do, you can express different sgRNA molecules. See, blue, red and yellow, these are different guide RNA containing sgRNA molecules driven by different Pol3 promoters. Now Pol3 promoters are not very big. Okay, so you can produce this tandem array of guide RNA encoding region. Okay, so what happens if this is the case? So you get 3 different kind of guide RNA simultaneously, and it will be loaded by 3 different Cas9 molecules. Okay, and this is what you want to achieve. So first strategy is clear to all of you? Okay, fine. No question? Yeah. These are separate unit only. This is, see, this is the guide RNA molecule. This is the guide RNA portion, 20 nucleotides. This is this green portion is the sgRNA scaffold. And then, although it seems green, but it is a violet. It is the termination poly encoding region. Okay, so these are individual units. Again, new promoter is there, so it will drive expression of this portion. Again, this is new promoter, it will drive the expression of this promoter. So you will get individual sgRNA. Clear? No question? If you have question, you can ask it right here. So next portion will be clarified. Okay, so should we move the next portion? Okay, so the next strategy is, now what we have done, we have produced individual guide RNAs, last strategy. This is called monocistronic units, but we can also produce polycistronic units, and this is strategy 2, and there is a protein called CSY4. Okay, this is CRISPR system of Yersinia 4. This is another protein which is also a endonuclease. Okay, and it has a very small recognition sequence. This C4 is its recognition sequence of 20 nucleotides, and it produces a ये जा सकता है नीचे, ये वाला पोर्शन, या ये रहेगा? ये नहीं जा सकता है. Okay, so you can see now, so this is the C4 region. It produces a hairpin loop, and this is the recognition sequence of this CIS4. Okay, this CIS4 protein, and once this CIS4 recognize it, it cuts it from here. So what you have to do? ये काम नहीं कर रहा. So in strategy 2, what you have to do, so instead of this vector, again you will be using a modified vector. Here in your vector, this cis encoding region will be there. It's not cis, this is CYS. Means it is CSY, okay. So it is difficult to pronounce CSY4, okay. CSY4, this is not cis, okay. CYS4, you have to have this in your vector, right? And your vector should contain this particular unit. See, here you have only one. Yeah, fine. So see, here you have only one promoter and it drives a polycystonic message. Okay, what is that? This contains, this is the sgRNA encoding region, this blue, red and yellow are the guide RNA, as you know, and this green portion is the scaffold, but they are flanked by this hairpin loop, which is the nothing but the C4 element, okay, which is the recognition sequence of this CSY4, okay. So what will happen? This is a polycystonic message, polycystonic transcript will be produced, and CSY4 will cleave it. Here you can see the scissor, which is nothing but this CSY4, and it will cleave it and produce individual sgRNA molecules, which will again be loaded into the different Cas9 molecules. So this is the next strategy. Clear? Any question? Yeah. Yes, CSY4 unit has to be there. CSY4, not CSY4, this recognition sequence of CSY4, this is called C4 unit, 20 nucleotides, okay. It has to be there. Huh? It is also endonuclease, okay. And one thing you have to keep in mind, that whenever you use this system, your vector must have this CSY4 encoding gene, okay. So it is a foreign protein, so you have to import it inside the plant system. And when you import a foreign protein, yeah. Yeah, it's not called restriction site, it is recognition site, okay. So you have to put it, this C4 units you have to put, see this black portions as those units, okay. Now when you put a foreign thing inside, foreign protein inside, so when you put this foreign protein inside, it has plus side also, minus side also, right? So there are reports that CSY4 produce cytotoxicity also if it is produced in large amount, okay. So there is a modification of this system, and now we come to the next strategy, this strategy 3, okay. And this is probably the most widely practiced multiplexing strategy, which is mediated by polycystonic tRNA gRNA transcript, okay. So what we do here, so you know that tRNA molecules, they are as such produced as polycystonic message. How many of you know that? Okay. So please keep in mind that tRNA, when it is produced naturally inside our system, it is produced as a polycystonic message, and there are enzymes inside every cell. So what are those enzymes? This RNase P and RNase Z. They are the naturally available enzyme inside the cell which process this polycystonic message to produce mature tRNA. Okay. Now people have utilized this particular strategy to multiplex. How they have done? So this is your this is your guide RNA portion. Okay, this is your promoter, Pol3 promoter, a single Pol3 promoter, as You know that in polycistronic, if you use a polycistronic way of processing, so you have only one promoter and one terminator. Okay. In between, you have to put, so this is your, this diamond is your 20 nucleotide guide RNA, and rest this rectangle, white rectangle, this is your scaffold. Now this has to be preceded by a tRNA unit. Okay. And tRNA encoding unit is a very small unit. Only how much it is, its length? Anybody can tell? Glycine tRNA, very common. It is about 75, 76 nucleotides. Okay. So that portion you have to add in front of this sgRNA encoding unit. Okay. So what will happen? You have a polycistronic transcript here. Okay. And in this polycistronic transcript, you can see there is a tRNA structure. This portion is your guide RNA, and this is your scaffold. So every sgRNA, that means guide RNA and scaffold, is preceded by a tRNA structure. And when this kind of structure are available inside the cell thinks it is my natural substrate. So it processes. Okay, using this RNase P and RNase Z, it processes. Right. And once it processes, it produces individual sgRNA, which is again loaded to the Cas9 molecules. So this is the strategy 3 what we have discussed. This we have already discussed. Okay. So this is the procedure. So here instead of this, you have to put this particular construct. Right. Clear? Any question? So here you don't need any foreign protein like CSY4 for processing. So no question of cytotoxicity. Okay. So there are two ribozymes. One is hammerhead ribozyme and ASDV. Okay. If you flank your guide sgRNA encoding portion by these two, now these two are cleaved. Okay. These two are cleaved naturally. Since ribosome, you know, ribosome are cleaved naturally. Okay, self-cleaving molecules. So it will be cleaved here and here. Okay, and the guide RNA will be, sgRNA will be produced. So you can make a polycistronic transcript like this, which is where your sgRNA is flanked by your hammerhead enzyme, hammerhead ribozyme and HDV ribozyme. Okay, and then you achieve the same result. Right? This is the strategy 4. Any question? Yeah. Come up again. Not really. There might be a little variation in frequency, but then again it depends on your gene also, and so people have used all these strategies. Okay. So there are many reports of comparison, and sometimes they correspond, sometimes they differ. So I have not presented that. I am not going to present that portion. I leave it that to you. When you do it in your work, you survey the literature properly and you choose your own strategy. Okay. So all of these are good strategy. Yeah. No, it's not much. See, your guide RNA is 20 nucleotides. It is one side, it has got sgRNA, means your scaffold is again 76, and this tRNA encoding unit is 77. So it comes around 170 around total. So one unit is 170. Okay. So if you want to multiplex also, so 170 multiplied by something. Okay, it's not much. Yeah, that is what I was telling her, na. So there are a lot of reports you can study. The lot of reports are there. They studied the comparative frequency of producing this knockout mutants using this strategy. So some reports they align with each other, some reports they differ from each other. So it is upon you. You have to choose what is your best strategy you think that will work. Okay, I will show you afterwards that we are using this PTG approach, polycystonic tRNA gRNA approach. Okay, but all are good. So these 4 strategies are based on Cas9. Okay, we everywhere we have used Cas9 only, right? But when we talk about multiplexing, we should not forget another enzyme. So there comes strategy 5. So you can change your enzyme altogether, Cas12a, and it has the natural ability to process polycystonic transcript. Okay, so people have used that also. So if you have Cas12, you need not to have anything else. Okay, it can process its own transcript, polycystonic transcript. So a little comparison, you can just go through it, I need not to explain also, so you can compare. Okay, so this is Cas12a, it is also called CPF1, and this is Cas9. Okay, now they are telling that it requires a single RNA molecule, and in Cas9 it requires 2. So you will be thinking that where we have used 2. No, we have not used 2, we have used 1 only, single guide RNA. But that is what we have done. This is CRISPR RNA and this is tracer RNA. We have joined it using a tetra loop. Okay, I think you must have been acquainted with this. Okay, and these have different PAM requirements. These two have different PAM requirements. NGG is the PAM for Cas9, and this is the PAM for Cas12a. Okay, let me quicken up little bit. Sir is already there. So now let us sum up. Okay, the different multiplexing strategies. Okay, so we have seen that we have Cas9 based multiplexing strategy, and under that there can be 2 approaches. Okay, one is integration of these monocystonic units, and you have seen it is done at sgRNA construct preparation level, and this is the approach. Okay, and under polycystonic this one, and it is this is processed at the sgRNA level, and there are again 4 different approaches you have already seen, right? So one is CSY4 mediated processing, one is tRNA mediated processing, another is ribozyme mediated processing. And then what I have told, that you should not forget about Cas12a, which has its natural ability to process polycystonic transcript. So this is the overall flow chart, I should say, for multiplex editing approaches. Okay, different multiplexing editing approaches. So as I have told little bit, now I will come to our work. We are using this particular strategy. I have already explained. Any question? Can anybody explain for me? Yes? Would you like to explain? Same strategy. Okay.
Specific multiple guide RNAs that could be 2 or 3 and more, and so whenever, like Pol3, whenever it will transcribe the whole construct, there are 2 enzymes, RNase P and Z, which cleaves the tRNA in order to process it, such that the guide RNA scaffold comes out. So different guide RNAs are formed, and now they can target their
different
sequence.
Genomic locations, right? So, so the process is okay, but how to make those constructs? Okay, this this PTG units. We call it PTG units because polycystonic tRNA gRNA assembly. Okay, so PTG. How we make it? So we use, if we want to make it in vitro, we need 3 different processes: PCR, overlap extension PCR, and golden gate assembly. Okay, so PCR you all know. PCR nothing to explain. So you have to have PCR, but in the PCR primers, see here is your, say this DNA 1 is your tRNA and DNA 2 is your gRNA. Okay, means this is DNA 2 is your scaffold, and your 20 nucleotides gRNA is here. Okay, now this is not the direct, this is the, see with reverse primer you take reverse complement of your guide RNA, and in next unit you add your direct sequence of your gRNA. So these red and this red and blue portion are complementary to each other, and this forms your guide RNA. Okay, so when you use PCR, so this kind of individual PCRs. Okay, so PCR 1 and PCR 2, so you get this kind of constructs. One is having this red portion, another is having this blue portion. Okay, now you go to overlap extension PCR. What is overlap extension PCR? The process is, it is like PCR only. Okay, but you use two templates, template 1 and template 2 simultaneously. Okay, and you use primers, only the flanking primers, this end and this end. You don't use these two anymore. Okay, so what happens? So this is a combined step. You cannot see it individually, but it happens inside the PCR. So at the first step, what happens? These blue and red portion are complementary to each other, right? So they are hybridized like this. Okay, and see there are 2 orientations. One is ending at 5 prime and another is ending at 3 prime. Okay, now DNA extends only from 5 prime to 3 prime direction. So if you use DNA polymerase, so it will extend this portion and this portion. So you will get this kind of initial template, and since you are using this terminal primers, so at the end of the overlap extension PCR you will get this bigger fragments ligated, which is the ligation product of this template 1 and template 2. That means your tRNA and gRNA, and in between you have that 20 nucleotide guide. Okay, clear? Now next step, so you have got TG unit. Now you need PTG. Okay, that means multiple TG units. How to do it? Can you do it by overlap extension PCR again? No, you cannot. Why? Because all the TG units have same tRNA and gRNA. So if you use primer, it will bind everywhere. So you cannot. So here you need another assay which is called golden gate assembly. What we do here? We use a restriction enzyme which is a type 2S restriction enzyme. Okay, what is type 2S? It has different means it has its recognition sequence and restriction sequence are different. Okay, and in between, see here, this is one of those type 2S. This is BSA1. So you see, you can, this is, it has some different recognition sequence elsewhere, and this is the restriction sequence, and when it cuts, it produces the sticky end containing 4 NNN residue, NNNN residue. Okay, now what is this NNNN? These are any nucleotide. You can put any nucleotide of your choice. Okay, so you can generate any sticky end you like. So you use this type 2S restriction enzyme. Okay, when you set your golden gate reaction, you use this type 2S restriction enzyme and your T7 DNA ligase, not T4, T7 DNA ligase simultaneously. Okay, and one thing only you have to keep in mind, that this n and nn should be different for all the units. So what will happen? It will simultaneously cut and join. Since you are using restriction enzyme and ligase simultaneously, so at the end of golden gate assembly you will get a ligated fragment, and I have only shown 2 here. If you have multiple, all this will be joined. Okay, so we will discuss in details during practical. I leave it here now. So this is the overall procedure. This is the primary PCR you have to do, then overlap extension PCR, and then golden gate assembly, and you get a polycystonic unit, PTG unit. Okay. Now you see, you have to use a lot of primers for that. Okay. Simultaneously, if you want to join three units, you need this much of primers. Okay. And for every see, this is the output of every reaction, and every reaction output is used as input for the next reaction. So all the primers have to be very coherent, and even if one primer goes wrong, your whole assembly will fall apart. Okay. So primer designing is bit complicated here. Okay. So you need, so this n is your level of multiplexing. You want how many guide RNA you want to multiplex. So if n is your level of multiplexing, this number of primers are required. Okay. If it is 3, then it is how many primers it would be? 14 primers. If it is 6, 26. Okay, understood. So the primer designing is bit complicated here. So we have developed a software. So, and recently we have got copyright of it. Okay. So this software helps you in designing the primers. Okay. And we have designed primers using this software. You can see we have joined 6 fragments, and this is the primary PCR, and this is the result of overlap extension PCR. So these 2 fragments has joined and produced this TG unit. Okay. So 6 TG units you can see, and after golden gate you have a ladder pattern. Okay. So if you are multiplexing 6 units, you should get 6 ladders. Okay. And the top of the ladder will be your BTG fragment. So you cut it here and clone it in a TA vector, TA cloning vector, what we have done it in. Okay. And then you sequence it and see that your assembly is all right or not. So if it is all right, then your job is, so this is the overall procedure we have discussed. So this is the PTG unit you have cloned here. Now you are ready for transformation. So this is, now you produce callus, agrobacterium, you go for agrobacterium mediated transformation or whatever transformation process you want to do. Okay. Then you develop plants finally. Okay. So this is the overall strategy. Now very briefly, sir is here, so I won't take much time. Very briefly, what we are doing, we are targeting a gene family in our lab. We are targeting a gene family which is called matrix metalloproteinase. Now the role of this particular gene family in plant is not known. This is basically a collagenase, and plant do not contain collagen. So what it is doing, we want to know. So what we are trying to do, we are basically want to functionally characterize, and we have seen that only 3 candidates are there which conform to classical MMP criteria. So this is very well known in case of vertebrates. It has sequences, the designated sequences, okay, consensus sequences, and this conform to the typical MMP molecules. Okay. So we are trying to knock these 3 out and also overexpress, and then try to identify the function, what they are doing. Okay. So for each of these units, for 3 genes, we have designed 2 guide RNAs each. It makes 6 guide RNAs, and then we have transformed, and I am only telling you the story of knockout. So we have generated 29 plants, and you can see this is the Cas9 specific PCR. So these are the number of positive plants, and when you get this positive plants, you amplify your target region. Okay. For what you are trying to do knockout. Okay, so you amplify that region and sequence it, and based on the sequencing result we got mutations. Okay, but only in case of MMP1. MMP2 and 3, they are somehow very resistant for mutation. So this is the second time we are trying, and both the time we have failed for MMP2 and 3. Only for MMP1 first time we failed, but this time we got mutations. Okay, so this first guide RNA, it produces more number of mutations, you can see, and you can see the effects of these mutations. Okay, so this is the structure of the original protein, and this structure is lost. Okay, some places where you have in frame mutations, you will still see that 9 base pair deletion. So this is in frame, so it has retained the structure. Okay, but otherwise it has completely lost the structure. So this is it, and we have got mutation for this guide RNA 1 portion also and guide RNA 2 portion also, which means our multiplexing is working. Okay, so I end here. So the last slide. Okay, I should thank properly everybody. So Vishwanathan sir, I think you have the pleasure to interact with him. Okay, so he is not only leader of our group, he is leader of genome editing in India. Okay, and these are our collaborators. Some of you already have interacted, some you will not. Dr. Rajkumar, he is out for his the Fulbright fellowship. He is in USA right now, and I have one ACRB project, and this Vanji was working with me. He has just left. Nazima is still working with me, and she will be taking the practicals today. And then we have got a ICR genome editing project, and these 4 people have joined newly. So this is our small group, and last of all, thank you everybody to be here and giving me chance to interact with you. Thank you. So we have this portion, but we will take, if we get time, we will take it in practical class. Okay, so let us listen sir now. Thank you.
So we would like to thank Dr. Mohan for nicely summarizing the multiplexing.
कौन सा वाला नीचे वाला नीचे वाला ये वाला ना
So friends, I have immense pleasure in inviting Dr. M. Reddy sir. Dr. M. Reddy is group leader at ICGEB. I think now he has superannuated, but he is still continuing as a Arturo Flasci fellow at ICGEB. Dr. Reddy is among the first ones in India to start genome editing work. I think the first NASF project which was granted, he was one of the important member of that group. And not only that, when I was at ICGEB, Dr. M. Reddy was our troubleshooter. Whenever we used to have any problem, we used to look up to him, and he was very prompt in solving our problems. So I have great pleasure in inviting sir. So thank you so much, sir, for agreeing to our request.
Good morning, all of you. I thank organizers for giving me the opportunity to interact with you in this training program. Our group is mostly involved in increasing the crop productivity, particularly in rice. Okay. So mostly I use the technology gene knockout in this training program, because gene knockout is not only the one technology, we can do another thing, because of the limitation of the time, I choose to explain the gene knockout, how I can apply in the rice crop improvement. My previous speaker already told to you how to introduce the tools into the plant system to edit. So I am not going to tell all those things again. So I will tell you how I use it for a crop improvement. I start with examples, and then I will explain how to do the things. So that in my talk generally, because the wider kind of audience, so I don't give any unnecessary technical details and boring you. So how I can apply only, I tell that technology. Okay, so that everyone will follow with me, so that I can tell you the things. So generally, if you introduce the Cas9 and guide RNA into any system, you can edit any plant system. To design the guide RNA, we use the softwares for a genome to unique for this gene of interest. Then the guide RNA will guide the Cas9 to the site of editing. Then you will edit the things. So we use a Cas9 expressing cassette and a guide RNA expressing cassette cloned into the right border, left border, and we introduce the tools through agrobacterium mediated transformation. So when you use the agrobacterium mediated transformation, it randomly introduced into the rice genome first. So wherever they introduced, the Cas9 will express as mRNA, then protein, come out and edit somewhere else. Similarly, the guide RNA can transcribe and come out and edit somewhere else. So even though the tools are integrated randomly, but they will come out and edit wherever you want to do the things. Okay. So general protocol is the same. What are the things? You make your cassettes into left border, right border, then use the agrobacterium to move into this thing. Then you generate a putative transgenic plants. Then use the Cas9 expressing using a PCR, whether the Cas9 is integrated or not. Those plants only you take for a further things. Then you sequence the targeted region and analyze whether the sequence is edited or not. Once it is edited, you remove the Cas9 tools out by segregation. Then you have a only edited plant without any transgenic footprint. This is the general protocol for everything. So I don't repeat everything whenever I am using the example. Okay. So I start with example, so that my aim is to improve the rice productivity. So I use a different examples. How can I produce a rice productivity? Okay. In this particular example, I am using it. If I increase the tiller number, I can increase the productivity. Okay. So this, if that is the strategy, then how I will increase the tiller number. this particular gene TB1 is negatively regulating the tiller number. If this gene is expressing, the number of tillers are less in the rice. If I knock out this gene, then more tillers can expect it. Because in all my cases, I told you that I am using a negatively regulating genes knockout, then getting the positive effect on the agronomic side. So here my tiller number increasing is the things. So this is my software design. This is a unique guide RNA for this TB1 gene in the rice genome. So then I use this and transform this, making this construct and transform the things. Then after that, when the Cas9 plant comes, so I design 2 flanking primers so that this much portion I PCR amplify and send it for a sequencing without cloning, doing anything. PCR product only you can sequence. Okay. So general protocol, what I showed in the graphic there, this way you get a putative transgenic plants, then first screen for Cas9 integration. Wherever the Cas9 positive plants, you use these 2 primers to amplify the target region. So this is the 500 base pair target region you will get. Then you send it for a sequencing using the internal primer to sequence it. So when the sequencing result comes, this is my guide RNA, this is my PAM sequence. After the PAM sequence, 3 bases it will supposed to cut. So when I am PCR sequencing the things, the sequence is clean up to the target region. After that, overlapping peaks will come. Why like that? If that is happened, out of 2 alleles, because every gene representing 2 alleles, you have edited only 1 allele, not the other allele. Then the 2 sequences comes from the edited allele and the non-edited allele. But here both are same, so that here the sequencing is very clean sequence. After that, overlapping sequences will come. If you get a sequencing result like this, then I am sure I have edited 1 allele at least. So how to make it a homozygous line? You self this plant and look for a homozygous line. When you self the plant, you will get a this is the control plant, this is the edited plant which I selfed. Then between this C to this C, 13 base pairs are deleted from this in the edited plant. When I put this 13 bases deletion in my computer and translate, the frame shifted, the gene is not functioning now, because in frame stop codons can come and the other than our expected amino acids, some other junk amino acids will come. So the protein is non-functional. So this way you can see, first you have identified a single allele editing. So now you are comfortable. I am edited single allele, I selfed. Then from the population, I segregated out first Cas9 out. The Cas9 minus plants I sequence. Then look for this kind of a both allele edited plants. Then in one go, I have removed the Cas9 and identified the homozygous for the both the alleles to be deleted. Similarly, when you do the things, another plant, I have deleted 24 bases. But when I put this 24 deletion and translate, the frame is same, come back to the same frame, because 24 multiple of 3 nucleotides, the frame is not shifted, only this much nucleotides, amino acids are deleted from the coding portion. The same, this is not knockout. The previous one is a knockout. Because my purpose is a knockout, I use my knockout things for an agronomic purpose. If I use my knockout lines, it produces 38 tillers. Generally, at the same conditions, non-edited plant gives the 13 tillers. So 13 tillers gives a 13 spikes. 38 tillers can give a 38 spikes. So you get a huge amount of productivity. See the things here like this. So increasing tiller number, you can increase the crop productivity tremendously. Okay. So, this is one example how I can increase the rice productivity. Another example I will tell you, the rice spike is a loose with a less number of grains. By using this DEP1 gene knockout, I can get a compact spike with more grains, and also it is semi dwarf also, so lodging effect and all those problems also solved. So, more grains will increase in the spike. Previously I increased the tiller, now I am increasing the grain number in the spike, okay, because of the compactness. So, I used a 2 guide RNAs here and transferred the same thing, the procedure is same. So, in this process I got a one base, I got so many things to do the things, because you get too many. For example, I am telling you, even a one base can change the reading frame, then it become a knockout. So, I am showing here like this, then this is a compact spike with more grains, so more yield comes compared to the control. So, this is another way of increasing the yield, within the spike increasing the number of grains. The spike size is same and the other things are same like this, but the things are improved. So, increasing the yield by tiller by increasing the grain number. So, now you can increase the spike size itself. So, how to do that? There is one enzyme called as cytokinin oxidase 2. This what the cytokinin oxidase will do, it inactivates the cytokinin by oxidizing it. If the cytokinin is oxidized, it becomes inactive. But cytokinin is essential for cell division and cell elongation and other things. If the cytokinin oxidase is there, it inactivates the cytokinin. Then the spikes, because this particular isoform cytokinin oxidase 2 expresses more in the developing spikes. So, when this is knocked out, the more cytokinin, functional cytokinin is available, then the spike grows bigger, more cell divisions, more elongation, and you get a bigger spike. Understand now? So, because I am inactivated the cytokinin oxidase, because cytokinin oxidase is not there, a functional cytokinin is more, because it is not oxidized and inactivated. So, if the cytokinin is more in the things, they can divide more cells and more elongations, bigger spike, more grains can form. So, use this thing, this is the guide RNA, this is the sequence of the cytokinin oxidase 2, then use the knockout. So, we deleted 2 bases, then the frame shifts, then it is not functioning. Then you see from these 2 bigger spikes, bigger panicles will come and more grains will form the things. So, this is another way of increasing the things to develop a bigger or enhanced number of grains in these things. So, another way of increasing the thing is I can increase the grain size, grain width, length, weight, all those things. So, increasing the weight also you can increase the total yield. So, this is another way of increasing the grain yield. So, here what happened, the GW2 QTL, which is a known, this is a kind of a ring finger type ubiquitin ligase. This ubiquitin ligase expresses in the rice husk and target the protein called expansin. When this ubiquitin ligase is expressed, it degrades the expansin gene. When the expansin gene is degraded, the spike in the husk size is small. So, only the smaller grains, smaller husk forms, and the endosperm also filled in the smaller things. If the if you knock out this ubiquitin ligase, then expansion is functional. If expansion is functioning, the bigger husk can form. So, bold grains will form. So, more endosperm will filled, more starch will be filled, and you can get a bold grains compared to the smaller grains. We have inserted a extra base here. Insertion or deletion of a odd bases changes the frame, then the gene is not functioning, and then you can see the things like this. So from smaller grains you can get a bold grains. The total weight of the yield can increases. Now I have shown the things increasing the yield by different methods. What you call as a tiller number, number of grains per spike, or bigger spike, or the bolder grains, like that. So another way of increasing the things is like this. So all the breeders increasing the rice productivity by breeding and bringing the bigger spikes and other number of grains, all tillers, whatever. So far I told you all those things. But all the elite varieties or the high yielding varieties suffering from one thing. Now what is the suffering? The grains on the upper portion, superior spikelets, are filled fully. The lower spikelets are not filled properly. So because of this, the quality of the rice, even though you increase the grain number, everything, you increase the things, yield increase, but these grains are not filled properly. When you are dehusking or milling the things for rice, they broke because the grain not properly filled. The broken rice will come. The head recovery of the rice will be reduced. So this is the major problem in all our high yielding varieties. Okay, why this is not filling properly? So if I fill these things also properly, then my yield can increase, or the quality of my rice quality increases. Because if it is not filled properly, they break in the milling time, and also in cooking time. The texture of this and texture of this is different. They may be very soft, they may be non-sticky, like that. The quality also, consumer don't like it. All these things can happen. Now I want to fill these grains also properly. So in a breeding things, people says I increase the yield, but this is the problem coming. Breeder don't know what to do with this. It's a very elusive for the breeder why this is not filling properly. This is not the genetic potential of the plant. They are not able to fill it. No, because these regions, the ethylene production is very high. When the ethylene is a plant hormone, gaseous hormone, when it is expressed more, it will inhibit the starch synthesizing enzymes, and this filling is very slow become and take a long time, but not properly filled. So if you can inhibit this, because the people have shown in experiments, when you spray some ethylene blockers or ethylene inhibitors, then they fill properly. Or if you increase the ethylene producers, they will deteriorate more. So when you can modulate the ethylene through genetic engineering, you can fill the grains properly. So ethylene is a very important enzyme, plant hormone. We cannot block the synthesis of the ethylene, but the ethylene, after the ethylene synthesis, lot of ethylene responsive factors, transcription factors are there. Those are the things further in the signal transduction they will do it. Now you have to identify the signal transduction, which ethylene transcription factors are involved in this ethylene production here. Those you block, not the ethylene synthesis you block. So then the grain filling will be okay. Because if you block these things, high ethylene means that ethylene transcription factor, that pathway is blocked. Then all the starch enzymes will synthesize properly and do the things. Generally what happened, this in another physiological way, in the spike, grain filling is a sink region, the photosynthesis is the source region to come. When the sink is weak, the photosynthesis is not transmitted completely here to the grain, then the accumulated photosynthesis in the flag leaf and other places, they inhibit the photosynthesis also. So even though they have a potential to fill this, ultimately they are unable to use that potential. So we knock out this ethylene things, then we can do the things, because we identified a one ethylene transcription factor 109 which is involved in this kind of things. We knocked out this gene by deleting some 11 bases or 11 nucleotides. Then the frame shifts. 11 is the odd of 3 numbers, so the frame shifts. Then it is not functioning. Then you can see these grains also fill properly in the knocked out line. So the inferior spikelets also fill starch properly, and the grain yield increases, the quality of the grain improves, the marketing value and the consumer preference are improved. So this is another way of increasing the rice productivity. So I have told you so many things now to do these things. Another way of problem is in rice. We are using the rice for a varietal breeding. Rice is a self-pollinated crop. We develop a variety and keep on using that. So after some generations, due to selfing, the quality also reduces in a further generations, the things. But if you can use the hybrid rice, I can increase the crop productivity. So this technology, hybrid rice, is not very popular in the rice. Why? The problem is, if you want to use a hybrid rice technology, you have to have a controlled pollination technique, because you need a large number of hybrid seeds. Not for an experimental purpose you can emasculate and cross, not like that. You have to produce a large amount of seed for a farmer supply. For that you need a regulated pollination you require. Mostly people use a cytoplasmic male sterile and a restorer line system, or they are using it. This system has a not a very good thing. First, out of the whole rice germplasm, the restorer line and cytoplasmic male sterile lines represent only 1% of the germplasm. So breeder has to do within that 1% for a hybrid vigor. So breeder has a restricted to use the parents, different combination of parents. For a best hybrid vigor he is unable to use it, because he has to use the male sterile line and restorer line and this kind of things. So because of this, breeder has a restriction to use it for a hybrid vigor. And another thing is this cytoplasmic male sterile restorer is a sick phenotype, because some defect in the mitochondria and its interaction with the nuclear genome together makes the plant male sterile. Because of this sick phenotype, the vigor for the hybrid is also reducing. And another thing, to introduce the other characters like abiotic stress tolerance or abiotic stress tolerance or some other things breed into that cytoplasmic male sterile line, then the property of the cytoplasmic male sterility is lost. So it is restricted to breed the new traits into the parental lines, also a problem in the cytoplasmic male sterile lines. Because of all these things, the seed production cost increases, but we don't see too much of hybrid vigor, so farmers don't prefer. So that's the one why we are not very successful in using a hybrid rice. We are still stick to the varietal. Another way of doing is a temperature controlled male sterility, or the photosensitive male sterility things are there, because under different temperature act as a male sterile line, and another temperature it act as a male fertile line. So two line system, this is good for you can breed anything. But when I talked to the seed companies, they said we are not dependent on this, because the temperature range is very narrow. The temperature fluctuations are too high in the environment. When we plant for a crossing, expecting male sterile become male fertile now, the whole lot we lost. And if one season we are not there in the competition of a hybrid seed supply, then in business we lose. So we don't depend on this. It is not a very reliable thing, even though theoretically you can say 2-line system is very good or this, that. Not possible. Okay, if this is the problem, I said I will make the any line into A line, any line into B line, maintainer line and restorer line, anything. So I can give a full freedom to the breeder, so that hybrid rice technology can be utilized for improving the crop. So many people said, oh, it is not possible. How can you make any line into A line, any line into B line in the breeding? I said, okay, I will do it. Then again some people objected to me. If you make any genetic engineering kind of thing, regulations comes into the problem and they don't allow us to use those hybrids. What can we do? Then I said, okay, everything fine. Whatever the hybrid you get without any genetic engineering component in that. So I will do that kind of things. I will show you how to do this. Okay. If we follow this, the breeder has a full freedom to cross any parent to any parent, get the full hybrid vigor, so that we can replace the varietal breeding into a hybrid technology. So how we do this? Suppose this is a mega variety. You take any variety, but for example I have taken this variety as a high yielding mega variety. I take this plant and knock out one gene, no pollen, because many genes are required for a proper pollen development. I can knock out one gene, then pollen development is stopped. Then it become a line, male sterile line. So now I can take any line, I can knock out this gene, I can make it a line. Now you agree that I can make any line into a line by knocking out a genome editing technology. Okay, I can successful in producing the A line. So then in the cytoplasmic thing, restorer line, maintainer line required. Now I need a maintainer line now. How can I make a maintainer line? Okay, take this knockout line. Whatever the gene I knocked out here, I put it back again as a transgenic mode. Okay, so because it is a dominant gene, even a one copy can restore the male fertility. But now this knockout line and the transgenic components are not linked. I knocked out at the original place, but when I put a transgenic plant, it is somewhere else. They are not linked together. Okay. Then I put brought the back into the male fertility. So again, I want to make the pollen is always in excess. So I want to make the 50% of the pollen sterile. Why I am doing this, I will tell you while we discuss the things. Okay. So I want to make so I don't require too much of pollen. So I require 50% of the pollen. I want to make it a sterile. So for that I use a Barnes gene expressed with a gametophytic, not sporophytic tissue promoter, gametophytic. It is expressed in the only pollen or pollen development thing only expresses the Barnes. So because it is a gametophytic promoter, the 50% of the pollen, because it is in heterozygous condition, 50% of the pollen is sterile, 50% of the pollen is fertile. Now to select that thing, I use put a herbicide thing, because I developed a very strong herbicide tolerance, glyphosate tolerant rice in my lab. Okay, I use that gene to develop these things. I linked all these things in one tDNA. Even though I put three components, whatever the gene I knocked out, I brought it back, and Barnes gene with a pollen specific promoter and a herbicide gene with a constitutive promoter. But all these things linked during my cloning system into one single tDNA region and transformed. So now you got a 50% male sterile fertile plant. This is my B line. Okay. Now I will explain how I maintain B line, how I maintain A line, how I restore the hybrid seeds. Okay. Yeah, correct. After knockout, I remove the Cas9 and other things out. So I am genome edited plant without any transgenic footprint. I take this seed and use it for a retransform with this thing. Now with the hygromycin selection, all those things are there in my tDNA. I made a transgenic plant now. My transgenic plant now is a 50% sterile, 50% fertile. This gene is there, original knockout is there, but I brought this gene back in the transgenic component. So because it restored the fertility, but because of the barnage, I made it a 50% sterile, and herbicide thing I use it for a selection later. Okay, now you got. Now I generated a line and I generated a B line. So now you people ask how you maintain B line, how you maintain A line, then how to use it as a hybrid seed production. First I will show you how to maintain the B line first. Okay, just take the B line and self it. When you self it, you can produce two kinds of things, A line and B line back. Because it is in a heterozygous condition, the pollen which is sterile is a transgenic pollen. Because of the barnage, it is not expressing. The pollen which is fertile is a non-transgenic. Okay, pollen is only non-transgenic coming. The non-transgenic is a knockout. From the pollen side, only one kind of gametes coming. Another gametes is sterile. In the female side, it can transmit. Okay, because it is a heterozygous condition, 50% are with the transgenic component, 50% are without transgenic component. So when they self it, you can produce two kinds of plants, with transgenic, without transgenic. Okay, without transgenic is A line. There is no transgenic component here. Transgenic component as a B line. So I got a A line and B line. The purpose of here is by selfing, I want to maintain B line only. So I can kill this by using a herbicide, because this herbicide tolerant is this, herbicide susceptible is this. So in the nursery bed, I can spray glyphosate, all the A lines are killed and only B line. From B line to B line I can maintain. So now you got how to maintain a B line. Now I will show you how to maintain the A line. Take the B line and cross with A line. As a definition, B line and A line are identical except male sterility fertility. So these are the same plants, made a transgenic for this purpose. So except that they are identical. So that way both A line, B line are identical isogenic, but only differ male sterility fertility. So now I use the B line to cross with the A line. The B line, the not transgenic pollen is sterile. It is giving only non-transgenic pollen. So A line is already non-transgenic. So non-transgenic pollen only given the pollination. So all the population get a pure A line, all female lines. So what the breeder requires, large number of female lines to cross with any line. So you produce maintain the A line in a large population. So after selfing, whatever you harvest, all are pure females. So you get a huge number. So breeder has a very happy to use this. So because this is a knockout of the pollen thing, any line you cross with, pollen restorative will come. So any line is R line for me. So breeder has a full freedom to cross with any line. So he can get the complete hybrid vigor. So these are knockout line. I can give a PCR marker for the breeder, and the B line transgenic also I can give. So this can be transferred into not only MTU 1010 whatever I start with, any line he can convert it into A line, B line by crossing using those markers. So breeder has any A line, any B line, any R line is available. So complete you can replace the varietal breeding to the hybrid seed set. So when the hybrid vigor is high, many advantages are there, because everyone knows hybrid is produce more compared to this thing, and mostly all the seed companies shy away from the using the rice for a business model, because if I give a rice, farmer can retain their seed and multiply their seed, don't buy the every time. Now the hybrid seed every time they can buy. So most of the seed companies jump into this model. Now they put their money like a cotton, one BT gene given, transferred into many varieties, and full advantage they got it. Now the breeders come into the things, the seed quality increases, timely seed supply available to the farmers, and the competition research will come into the private sector. All the rice crop improvement can happen using this technology. So we can do the genome editing technology and other combination of technologies. We can convert any line into any A line, any line into B line, so that we can replace the low productive varietal with a hybrid seed technology. Okay. So now I showed you so many ways I can improve the rice productivity in the country, so we can beat the China very easily. Okay. So you can cross with any line, you can restore it back. So at the end, what you want to see is this is the non-transgenic. Any line, normal line is a non-transgenic. Your hybrid is a non-transgenic. So no regulation. Only B line maintaining is the seed company will do at their own site. Otherwise, everything is non-transgenic. In the farmer field, everything is non-transgenic. The hybrid seed is non-transgenic. It is not regulated. So far, I have seen how to improve the yield. So another way of improving the yield is how to prevent the losses. If you can prevent the losses, the sustainability of the productivity will maintain. So lot of abiotic stresses and abiotic stresses are making the potential yield losses into the other way. I am showing here to develop a drought tolerant plant. So we are using some ubiquitin ligases, because these days ubiquitin ligases are shown involved in the lot of abiotic stress tolerance, because mostly previously try to use the overexpression of gene and do the things. So another way of knocking out is when the stress comes, post translational protein abundance you can regulate by ubiquitin ligases. Particular protein already synthesized, transcribed, that protein we have to reduce now because of that, to adjust to the abiotic stress. So that time ubiquitin ligases plays the things. After post translation, you want to regulate the protein abundance, the ubiquitin ligases do the things. So a lot of ubiquitin ligases are available in the plant. Each has their own clients. Only those plants only they can proteins only they can ubiquitinate like this. This particular ubiquitin ligase was knocked out when we one base was inserted here, one base A or T is inserted, or again 21 bases are deleted. 3 kinds of mutations I am showing here. One base insertion is a knockout. The 21 base deletion is came back to the same frame. Okay. Now I take those seedlings and test it for a drought tolerance. Okay. This is the control plant. This is the A inserted, T inserted, whatever knockout. Then this is the 21 deleted, but frame shift back into the thing. After going this much stage, I stop watering. I stop watering up to 14 days. I showed here 12 days only. I went to the 14 days. So after the 14 days, when I rewatered back, so all the knockout lines recovered back almost 90%. All the other things were collapsed. So even though I am showing at a seedling stage, by knockout of this ubiquitin ligases, particular ubiquitin ligases made a drought tolerance. You see here. So we studied something why this is coming like this. Mostly we have observed is the transpiration is very less in these plants, knockout plants, compared to the other things, because the control plants lose a 28 ml per day in our experimental model. Then the knockout plant only lose 16 ml. So because of the less water loss, the water retained in the pot maintained for longer time and delay the onset of the drought or drought situation. Okay. For another reason, that's the one way. So then we studied the number of stomata. Why the transpiration is less? So almost 50% of the stomata is reduced here, but that reduction of 50% of stomata has no effect on photosynthesis. Then we studied the roots under these conditions, because under the water irrigation when we stop, because each plant is losing water at a different concentration, so they are not coming into the same stress at a given stage. So we use a mannitol or a PEG solution to bring them at a same stress condition. At that kind of situation, what happened, the roots are long, more thin, and other branches are more. And when we studied the anatomy of these things, the number of xylem vessels are more in the control, less here, and the lumen of the xylem also narrowed. Okay. So all these things what it has done happened because of the more branching, more thin and long roots. That means with the same amount of energy or a carbon input, the specific surface area is increased in the root system, so that they can forage larger area for a water absorption and supply to the things. Because of the narrow leaves and the less lumen, because of the less lumen, when the water is limiting, transpiration happen, the root is not supplying that much water. Finally, the water column will break. A cavitation or embolism can happen. Air bubbles can enter into this. So those things, when you rewater, the water column is broken, it won't recover back. Because of the narrow xylem, it maintains the cavitation resistant. Then when water gives, the water column restores back and recover back at almost 90% back in these conditions. Because when you grow the plants together, the height is reduced. You see here. The reason for that is when the xylem lumen is narrow, the axial conductivity is resistant, because they cannot lift the water very high. To compensate that, the plant is reduced the height and stomata reduced because of the restricted water. Because of this, water use efficiency of this particular thing is it loses less water, produce almost similar grains. So because of that, water use efficiency is increased. Whether it is a really drought tolerant or not, to see that we have done both the plants grown in the same pot. Because when the different pots, one pot is losing more water, one pot is losing less water, to maintain them is difficult. Both put the things here. The control plant was almost wilted, the other is still turgid and green and growing. Because of the two reasons. The evaporation demand is less in those plants because of less stomata, less transpiration, and the roots are more fibrous and thin and spread. They can still extract water from the dry soil. Other plant not able to do the things because its demand is more, supply is less. But in other cases, demand is less, supply is more. So they can survive in the same pot also like this. One way it is delaying the onset of the drought situation and saving the water, and also resistant to that dry soil. From even the dry soil, it is able to extract water and able to survive. So like this, we can make a drought tolerant Because of this, you can save the losses of the productivity. Okay. In another example, I want to show you here, because the lot of changes in the climate these days, unpredictable. To match to the this unpredictable climate change, I have to make a resilient rice plant. So most of the cases, when the trehalose is accumulate in the plant tissue, they can survive drought, they can survive cold, they can survive heat, all kinds of things they can do the things. Because everyone knows if you increase the trehalose in the plant system, they can do this. Because many people tried, probably Anil also tried when he is in ICGB to increase the trehalose, but not successful. The reason for that is when you these genes, two genes are required from to make the trehalose. When the trehalose is made, there is another enzyme called trehalase that is degrading back into glucose. So effectively you are unable to increase the trehalose content in the plant. You are trying to improve the overexpression of these genes, but this is degrading the whatever the product coming. Effectively the trehalose is not accumulating that much. So now I knocked out this gene, degrading gene. See, this is the trehalase encoding gene. Then we knocked out, almost 360 nucleotides were lost. Using this plant, using this knockout plant, you can still get a tolerance. But if I use this plant to overexpress now these genes, then more trehalose will come. So when more trehalose comes, your plant can dry in the drought, but it won't die. Like this Selaginella, you can keep this dried plant in your cupboard for 1 year, no problem. Then you sprinkle little bit water, after 1 year it will grow from there. Because what happened when the trehalose accumulates more, when the water is removing, then before the cell will collapse, the trehalose become like a glass. It is frozen like a glass. So in the frozen glass, the cells are still living. They are not died. Because of the frozen glass, they are protected. When you sprinkle the water, the glass melts. Then the cells start working. So like this, 1 year you keep in your cupboard, then you sprinkle water, still grows. Like that I want to make the rice. Like that, so that any heat comes, cold comes, so my rice won't die. It will come back. So that's what I am trying to do this. So far I have knocked out. I am trying to overexpress these genes. Probably I will achieve these kind of things. I showed you two examples for abiotic stress. How can I prevent the crop losses? Okay. Another thing is abiotic stress. So biotic stress, a virus, bacteria, something can happen. What are the things? Suppose I am using a virus here. So I want to make a virus resistant plants. When the virus infects the plant, virus uses the plant machinery to translate its machinery. So it is using the host machinery for this purpose. When the host machinery has a redundancy, suppose I use a translational elongation factor thing. There are so much redundancy there. So anyone can support the plant. But virus when evolved, it can use either one isoform or another isoform. Suppose you knock out a isoform which is virus is using, then virus cannot multiply. But plant has no problem. So if you knock out like that, whatever the isoform the virus is using it for its machinery to translate or transcribe, then the virus cannot multiply. You are making knocking out that isoform, you are making the virus resistant plant. Agreed? The other isoform will compensate for the plant purpose, but virus cannot grow the things. You can make a virus resistant plants like that. Is possible. Another way of the thing is, when the any pathogen enter into the plant system, then it expresses some proteins which go and activate the susceptible genes. Susceptible genes are nothing but when they are overexpressed susceptible genes, the innate immunity of the plant is reduced. When the innate immunity of the plant is reduced, the pathogen happily live on the living cells and feed. So they will do this kind of things. Now you know what are the susceptible genes. So you knock out those susceptible genes so that the pathogen cannot induce those genes for their survival in the system. Otherwise, plant innate immunity can attack the invading pathogens and prevent the things. This is another way of suppressing the pathogens and preventing the production losses by biotic stress. So because of the time, I am not explaining too much on that, but as a logic you understand now, the susceptible genes you knock out, then the pathogen cannot induce them for their own favor, so the plant system innate immunity can control them for further losses. Now I show one or two examples because my time is over. So instead of crop loss, crop improvement, suppose this phytic acid is a huge amount of, it's called as an anti-nutrient for us, but it is good for the plant to protect from many things. Because how to prevent this thing, because the phytic acid is a one inositol ring with the 6 phosphate groups attached it. Because all the 6 phosphates, each phosphate has 2 negative charges. So 6 into 2, 12 negative charges are there in this molecule. These 12 negative charges are chelate all the positive minerals. So we feel a mineral deficiency, micronutrient deficiency we will feel because of this. We are eating enough micronutrients, but we are not assimilating because of this anti-nutrient. Because this is in a large amount present in the cereal grains and pulse seeds everywhere. So because of this we have a problem. So we can knock out this, because in many breeding situations they tried it. Actually what happened, when this phytic acid pathway, you see the things, many of the intermediates are essential for the plant growth. So when you take the mutation of here, the plant is defective. If you take the last genes, the phytic acid is not formed, but the last gene mutation is not affecting that much to the plant development. So you can knock out this gene, the phytic acid is not formed, all the intermediates are formed, so that the plant is nutrient free. So that particularly in the rice it is not considered too much of phytic acid, because we polish the things, the phytic acid is removed from the aileron layer. But these days the rice bran is fed to the animals. If it is a ruminants, they can digest the phytic acid, but non-ruminants, they have a problem of these things, this kind of thing. Also these days phytic acid is, this rice bran is used for oil extraction. So when the phytic acid comes into the oil, again problem for us, that kind of things are there. So if you knock out this gene without affecting the plant development, and you can remove the phytic acid using this kind of situation. Another thing is a low glycemic index rice you want to make for a diabetic patients, or so many other things. Resistant starch has so much help for a colon health, colon cancer can be prevented, so many things can be happened like this. So here rice is a polymer of contain amylopectin and amylose. Both are the polymer of glucose only. One is a branched polymer, another is an unbranched polymer. If the unbranched polymer amylose content is more, then it is a low glycemic index, because our enzymes release the glucose from one end slowly, regularly. If too many loose ends are there, this can release from glucose here, all loose ends. So the quick glucose in the blood increases. So you can reduce the amylopectin content and increase the amylose content. Amylose content you increase is a hard rice. You can chew, get the taste. If it is a too much branch, is a soft rice, sticky rice, but we don't prefer. We like biryani or whatever the thing. They should be, each grain should not touch each other, like that kind of thing. So we should reduce the branching. So you can take out the starch branching enzymes, knockout. So amylose pectin content reduce and amylose content increases. So you can change the quality of the rice and glycemic index of the rice also. Okay. Because these days, previously rice bran was as a byproduct given to cattle, but now we are isolating a rice bran oil. So that is a more healthy oil these days. So they use the things here. Generally all seed oils has a omega 6 rich. The omega 6 is not good for us because we have to eat the omega 6 and omega 3 in a particular ratio. If you are eating a proper combination, you are guaranteed without any modern diseases and 100 years you can live properly. Okay. Because we destroyed the omega 3 completely from our food chain. So all the vegetable oils are rich in omega 6. The omega 6 is a inflammatory reaction. Omega 3 is a nullify the inflammatory reaction. Sometimes what happens, inflammatory reaction is required for the immune system to boost, but at the same time it should be countered by the omega 3. Otherwise all inflammatory reactions gives the all these modern diseases. Okay. So we are suffering from the omega 3 and or suffering from the too much of omega 6. So to decrease this thing, for cooking oil purpose omega 9 is preferred because this is shelf life and everything is thing. If the omega 6 is more, rancidity of the oil and the cooking products also smells after some time due to rancidity of the double bonds. So if you engineer this delta 12 desaturase, then it stops at omega 9, it won't convert it into omega 6. So if you increase the omega 9 or a monosaturated or high oleic acid containing cooking oil, the rice bran oil quality will improve very good, and it is preferred for health, and it reduces the disease burden so much because it is preventing the omega 6 formation, because we are suffering from the omega 6, too much of omega 6 we are eating. So that can be engineered by knocking out this. So I will stop it here. I showed you the different ways we can increase the productivity and other things, because this has I showed only one thing, how to knock out and use the things for our purpose, because CRISPR can be used for so many other things also. I showed only how we can use the knockout only, because you can imagine whatever is possible with a genome editing technology. So you can ask me any questions you have. Huh?
So how can we cope with that?
First of all, when I made my TB1 gene has a very high yield because of again a lodging problem. If you want to do your own way of doing the things, when I knocked out at different places, if you put your guide RNA positions differently, allelic series you can develop. Some allelic series, the bad effects can reduce and the good effects still you can maintain. Suppose you have too much of tillers, that much tillers may not required probably. You can reduce a little bit number, but the yield you can increase as much as possible. Lodging you can maintain the thing. Like that allelic series these days shown that the bad effects can be removed like this. Another way of thinking is by gain of function you can get a lodging tolerance. So I showed only the loss of function. So you can edit a gain of function gene and bring the tolerance back, or there strong column varieties are available, you cross and bring your mutation into that and bring the strong column and make a lodging tolerant. These are all possible because it's under your control.
Sir, can genome editing be used for site-specific gene expression? Changing site-specific gene expression?
For example,
Like ethylene you have told, no sir. We have to reduce the ethylene production so as to increase the quality of grain filling.
so if you understand the pathway which is the gene involved in that, that you knock out, because ethylene responsive factors in combination with the ethylene are controlling the trait. Because so many ethylene responsive factors are there, they are involved in all developmental process. You identified for a grain filling purpose only, so that isoform is not showing any bad effect on the other development, only helping here. You can knock out like that, no problem.
Okay. Sir, usually guide RNA will be inserted, no sir, for genome editing. Why do we use 2-3 guide RNAs?
To increase the probability in your favor,
Okay.
because so far all the literature says all guide RNAs are not equally efficient, because in the native conditions the DNA is not a pure DNA. It is combined with so many proteins and packaged into a chromosomes and other things. When the Cas9 goes, this guide RNA may be available, that region is available for a thing or not, we don't know the thing. That's why we take 1 or 2 more guide RNAs. Any one will help you to knock out. So that way the probability increasing in your favor. That's all.
Sir, multiple genes can be edited at the same time using different guide RNAs?
Yeah, correct.
Yes.
Because Cas9, whichever the guide RNA it interacts, that guide RNA will direct the Cas9 to that place. So you can edit many genes simultaneously, no problem.
Thank you, sir.
Okay.
Any line, and we can convert any line into any line, means A line into R line or B line.
Yeah, I showed you any line I can convert into A line, any line into B line.
So instead of say genome editing, can we go with the mutation breeding?
See, if you ask a different question, I didn't tell you here. Suppose so far which gene to edit, then you are editing. If I don't know anything, I want a particular phenotype, what can I do? I can do. Okay, how I do? I take the DNA repair system genes to knockout. When the frequency of the mutations will increase. Previously, what is the mutation breeding? One time you expose to gamma rays or EMS, then within that you try to look for your mutant. Instead of doing that, I mutated the repair system. The efficiency gone. It may not be lethal, but it's producing too many mutations. That time, if you want a drought tolerance, you create a drought and grow your seedlings. Some seedling will start growing in the drought conditions. You don't know which gene is mutated. A lot of mutations are coming. You give the screening different things, you get things. You take that plant and take it to further drought. It's like in a bacteria they do a directed evolution mutations, like that in the plant you can do the things. You don't know which gene to mutate, but you can screen. You can make a one bag full of rice seeds from your mutant plant, and somebody wants a heat resistant plants. Increase the heat and grow there. Some mutants start growing. Take that and increase the heat. Further selection can happen. So like that, you don't know which gene to mutate. I have a factory of mutants, mutation continuously happening. I use my different selections. If any lethal mutation, that will go away. Any non-lethal mutation, you will see the things. Whenever you got your required phenotype, you segregate out this gene, mutated gene out. So your thing is stable. So after that, to publish, do all these things, what is mutated, not mutated, you find it out. But I got, without knowing the thing, I am only made the mutation factory for me, and I am screening whichever the thing. Somebody want a herbicide tolerance, somebody want a high yielding, somebody want a early flowering, whichever the thing you select. The mutants are continuously making, not one time exposing to gamma rays and doing the things. This way you can do it.
Thank you, sir.
सर जब वी क्रॉस या सर you cross transgenic and non-transgenic, on which generation you got the homozygous non-transgenic line?
Most of the time, in the first time when you do the thing, one allele editing is a common thing. Okay, so once one allele is edited, you self it, you automatically you will find the 1/4 of the population you should get a homozygous for this.
And sir, one more.
Sometimes, rarely, how it happens, I also don't know how to explain the things.
Yes.
So homozygous line comes in the first generation itself also. But how it happens, I am unable to explain to you. Okay.
And sir, the transgenic cross with non-transgenic, is it come in regulation proper? Means in India, transgenic is in banned condition.
There are more sick people in the regulatory system.
Yes.
They will think transgenic is the problem. Okay.
Yes.
The current problem, we are killing the mother earth by high input agriculture. We are destroying the things. If you use a transgenic things and reduce your pesticides, reduce your high input fertilizers, your earth will be survive for long time. Even today, previously 10 kg of urea is enough to give the thing. Soil is that much spoiled. Even you give a 30 kgs of urea, the 10 kg effect is not coming, because it is not able to absorb, because you destroyed the soil so much. After some time, soil is a sterile thing, no plant can grow like this. Because tell your regulators, we are killing the things by using high input agriculture, not by transgenic.
Okay.
Because what is the problem in transgenic? One small piece of DNA I put it, even it's a cisgenic I can put it like that. What happens? Nothing can happen. When I in the morning I ate so many other things. I ate chicken genome, rice genome, or whatever. So many genomes I ate. Nothing happened to me so far. So if I put a small DNA piece into that, there is a sick people sitting there and preventing the things. Nothing else.
Okay. Thank you, sir.
Sir, in case of the triglyceride gene edited plants, triglyceride gene. Sir, triglyceride. Sir, when there is no enzyme triglyceride, that time over accumulation of the triglycerides will be there. It will cause any effect to that plant.
So theoretically, at present, if I overexpress triglyceride, what the agronomic problem comes, I am not bothered at present. Can I make this plant resistant to all kinds of weathers? Is my present thing. When I achieve that thing, the other problems I can solve. Because if you imagine too many things and worried about things, because should not worry. If somebody can do it, I can do better. Like that mentality you start. So we will get it.
Sir, it will tolerate to cold also. You told the heat, cold.
Yeah, because many examples were previously told that the triglyceride accumulation, protect from the cold, protect from the heat, protect from the drought. All these things are shown in the literature. Only we are unable to increase this to the higher level because of the triglyceride enzyme. So when you knock out this, now you start improve the things, it will happens. Because all the genes required for the triglyceride synthesis are present in the rice, but they are not expressing constitutively, because they are still expressing in the pollen. When the pollen is detached from the anther and going to the another stigma, that time the desiccation protection required. In the pollen grains, triglyceride express. When the seed is maturing, desiccating in the seed. All the endosperm, other things are non-living. Only the embryo is living. When the embryo is living and desiccated so long, and 1 year you can keep the seed like that, still protecting, because the trehalose is expressing there. The trehalose is protecting that embryo. When the water comes, trehalose melts. Then this is the 1 year, that much desiccation, how the embryo is still maintaining because of the trehalose. Now I want to express this, all the tissues, so that all the tissues can protect, like the embryo after desiccation. After desiccation it will dry, but it won't die. That's what I am trying to say, the things. It will start from there.
Sir, actually when we knock out the triglyceride enzyme producing gene, there will be no triglyceride enzyme. It will affect any other function or only for this
So when we knock out the things, it has no bad effect. My plants are still okay, all the things okay. But when I overexpress, what happened to the plant, I also don't know. That problem I will solve. Let first I achieve what I want first. Okay?
Okay, thank you, sir.
Sir, I have doubt on my research topic. I have selected a gene which control secondary metabolites, sir. I am expecting that actually generally secondary metabolites like isoflavones will give resistance for fungal diseases. So I am using that gene like for knocking out to impart resistance for viral diseases. Can I expect resistance for viral disease?
No, just now you said the secondary metabolite is essential to protect from the fungus.
Generally biotic stress and abiotic stress.
No, by knocking out the pathway wherever the thing, you are increasing the metabolite or decreasing the metabolite?
Increasing the metabolites.
Increasing the metabolite automatically should protect from your pathogen.
Can I expect for viral diseases?
Depending upon the things, whatever the things, if the innate immunity increasing or the viral machinery which is using the plant machinery, I am knocking out. I am making it a generalized there. So because every virus can use the machinery of the plant only. So if I knock out those isoforms which is used by the virus, then virus resistant comes. But secondary metabolites, I don't know what the virus can affect or whatever the thing.
Okay, sir. Thank you.
So, if there is no question, I would like to thank Dr. Reddy for delivering an extremely useful talk to all of us and answered your queries. Token of appreciation and love to sir.
Good morning, students, trainees. So after this very enlightening morning, now this is the time for tea break. So you have the tea break, and after the tea break, all of you, that is the 35 trainees, shall proceed for today's practical class. And today's practical class is going to be organized at Discovery Center. So Discovery Center is the hub of all NAHEP related research activities in this institute, and it is located right next to my division in Division of Plant Physiology. So the bus is waiting outside. Quickly have the tea, board the bus, and be at the Discovery Center. Dr. Soham is waiting there to conduct your practicals there. Please.